Alright, so good day my dear grade 12 students. Welcome sa aking recorded video discussion. So last time, history ng molecular genetics yung pinag-usapan natin. So the purpose of that was to show you that like every other discipline, genetics has its humble beginnings. From debates and bickering of prominent scientific figures to milestones and new discovery that we have right now. So the fact that we are discussing and talking about these concepts in our school is a celebration and appreciation of their contribution. So the central dogma of molecular biology, to simply explain it, so it is the flow of genetic information from DNA to RNA. And from creating a functional product out of the genetic information, which we call now as proteins. Okay. Now, bakit kaya tinawag na dogma, yung central dogma of biology? Di ba kapag naririnig natin yung salitang dogma, ang lumalabas sa isip natin is turo ng simbahan, gano'n, doktrina. Okay, the church dogma. Okay. So, pero ano ba yung definition ng dogma? Okay. So, ayan. Dogma are tenets, they are principles, they are sets of rules that are set up by authorities. It cannot be erroneous. Okay. So, dogmatic yung approach kapag ganun. Now, similarly, ganun din sa central dogma of biology. It is not erroneous and it is a compilation of the works and toil of different scientific authorities. Yung mga na-discuss natin nung last time, they are the foundations of the central dogma of biology. Yung mga scientists na yun, the men and women of science. Sila yung naging pundasyon para magkaroon tayo ng central dogma of biology. Okay? Para ma-discover natin yung mga processes involved sa central dogma of biology. Okay po ba? Ayan. So, ang central dogma of biology, meron tayong tatlong processes. Replication, transcription, and translation. Okay? But before that, let us have a quick recap. So, itong diagram na to, I'm sure na yung iba sa inyo nakalimutan na yung ibang concepts related dito. So, this is a diagram of cell cycle. So, we have gap 1 wherein the doubling of extracellular contents happens, rapid cell growth, ano pa ba? So, yun nga. So, sa gap 1, nagkakaroon ng growth. So, nadodoble yung extracellular content, except sa chromosomes or the genetic material. Kasi, duplication happens in S phase. So, sa S phase, nagkakaroon ng duplication ng content, ng genetic content. Okay? Then, sa gap 2, nagkakaroon tayo ng rechecking ng chromosomes na nadoble dito sa S phase. Okay? So, yung mga duplicated chromosomes, kailangan na lang mag-undergo ng rechecking, proofreading, and repair dito sa G2. Kasi, kapag nagka-problem dito sa G2, hindi ma-check, magkakaroon ng harmful mutations. May it be at the DNA level or at the chromosome level. Okay po ba? So, syempre, meron tayong mitosis, the cellular division itself. And G0, the cell cycle arrest or simply the cell stop dividing. Okay? So, ganun. Now, yung unang process na pag-uusapan natin is replication. And ang replication, nangyayari siya dito sa S phase. Okay. So, dyan sa S phase, nangyayari yung replication. Okay. Now, we have different theories of how does replication occur. But among the three theories, the accepted one is the semi-conservative replication. Okay? Now, what are the other two? We have conservative and we have dispersive. Okay, now let us explain first the conservative. So in conservative replication, in this model, DNA replication results in one molecule that consists of both original DNA strands and other molecule that consists of two new strands of DNA. Okay? So yun po, yun yung general description sa kanya. Okay? Now, nakita nyo, itong sa GIF. So, we have a DNA here. Okay. Now, itong DNA na to, mahati siya sa dalawa. So, ito yung magiging templates. Dalawang template. Old strands. Okay? Meron tayong dalawang old strand. Ngayon, out of these old strands, magkakaroon tayo ng complementary strand or mga bagong strand. Okay? Na ngayon, after magkaroon ng complementary strand, yung dalawang old strand, mag-join ulit sila, forming one DNA copy na binubuo ng old strands, purely old strands. And yung mga complementary strand na nagawa dun sa old template, sila yung magsasama. Okay? So sila yung isang copy na binubuo ng entirely new strand. Okay ba? So ayan. For the dispersive model, dispersive replication is the second model of DNA replication. So the model produces DNA helices that contain a mixture of old and new DNA. Thus, each new strand in the helix is a patchwork of old and new DNA. In simple words, all strands in the DNA helices contain alternating parental and new DNA segments, as explained in the model here. So scientists believe that DNA only copies itself for short chunks at a time to produce an alternating pattern of DNA. Okay? Now, we have the semi-conservative replication. In this model, the two strands of DNA unwind from each other, and each acts as a template for synthesis of a new complementary strand. So this results in two DNA molecules with one original strand and one new strand. So in here, meron tayong DNA. Ano itong DNA na to? Mag-isilbi siyang template para sa ating complementary strands. So we have two template strands, and out of these template strands, the complementary strands were bind with the old ones. Okay? So kaya, sa semi-conservative model, ang kada copy ay binubuo ng isang old strand and isang bagong strand. Okay. Yun yung pagkakaiba nila with semi-conservative and conservative model. Conservative model, yung isang strand sa conservative model ay entirely old. Yung isang strand naman is entirely made up of new strands. In semi-conservative model combination, meron tayong old strand, meron tayong new strand sa isang DNA. Okay po ba? Ayan, napaliwanag na natin. Okay, next. For the first part of replication, the initiation, very self-explanatory. Okay, diba? So initiation means start. So what are the mechanisms para mag-start yung replication? So, ito muna ipapaliwanag muna natin yung pagkakaiba muna nung prokaryotic and eukaryotic replication. So, para sa prokaryotic, meron tayong isang origin of replication. Okay po. Para naman sa eukaryotic, meron tayong multiple origins of replication. Ayan. So, in here, sa eukaryotic cell, makikita nyo, dito sa example, meron tayong tatlong origin of replication. But in reality, masyadong mahaba ang DNA ng eukaryotic cells compared sa DNA ng prokaryotic cell. Sobrang haba ng DNA ng eukaryotic cell, hindi lang siya limited sa tatlo. Yun ang tatandaan natin. So, multiple origin of replications ang meron ng eukaryotic cell. It is just that, in this example, ang pinapakita natin ay tatlo lang. Okay? So, yun yung dapat clear sa atin. Okay po. Now, yung pareho sa kanila, meron tayo ditong replication forks na tinatawag. Yung replication fork, yan yung lugar kung saan nag-i-insert ng mga bagong nucleotides. Okay po? So, meron tayong isang replication fork dito, meron tayong pangalawang replication fork dito. Okay. So, habang nagkakaroon ng mga bagong strand, nagkakaroon tayo ng tinatawag na bubble. Okay. Yung bubble dito sa prokaryotic cell ay isa lang, syempre. Pero yung bubble na meron sa eukaryotic cell, multiple counts. Okay? So, more than 3 bubbles actually na meron. Okay po. So, yun yung pagkakaiba ng prokaryotic initiation from a eukaryotic initiation. Okay, tatandaan nyo yun. Ayan. So, makikita nyo naman dito sa ating example, di ba? So, ito ay isang prokaryotic cell. Kasi meron lang siyang isang bubble. So, ibig sabihin, meron lang siyang isang origin of replication. Dito naman, sa eukaryotic cell, bilangin natin, meron tayong 1, 2, and 3. So, meron tayong more than one replication bubble. So, indeed, this is from a eukaryotic cell. Okay. Now, to make replication possible, we have enzymes and proteins na nag-act and tumutulong para mapadali yung process. Okay po? So, halimbawa, we have here a DNA strand. So, tingin muna dito sa embedded na narration. So, for example, we have here a DNA strand. And ang DNA strand is coiled, di ba? So, coiled yung DNA. So, prior to that process, syempre, ang structure ng DNA ay supercoiled. Ayan. Supercoiled yung DNA. Okay. Now, ang ginagawa ni gyrase or ni topoisomerase 2 ay ina-uncoil niya yung supercoiled structure ng DNA. So, from the supercoil, ina-uncoil niya. Okay po. So, yun ang function ng gyrase or ng topoisomerase enzymes. Okay? So, tatandaan. Now, after that, meron naman tayong tinatawag na DNA helicase. Ayan. So, ang DNA helicase naman, ang ginagawa niya ay i-unzip yung DNA strands. Okay? Habang ina-unzip niya yung DNA strands, syempre, yung tension dito sa other part nitong DNA na hindi pa na-unzip ay nag-increase. So, mas nagiging coil yung part na to. So, dito sa mga part na to, mas nagiging coil. Kaya, si DNA gyrase or si DNA topoisomerase, patuloy niyang ginagampanan yung role niya na i-relax or i-uncoil yung DNA. Okay po ba? Ayan. Now, itong mga nucleotides naman na to prevent them from zipping again, we have the single-stranded binding proteins. Or, tawagin na lang natin silang SSBS. Okay? Now, the SSBS proteins hinders the nucleotides, the complementary nucleotides from having contact with each other. Kasi kapag nagkaroon ng contact ng mga yan, mag-zip ulit. So, magdidikit ulit. Sasara ulit yung binuksan ng DNA helicase. Okay. Now, we have here another protein which is the primase. Now, primase, the function of primase is to insert new nucleotides, at least the initial nucleotides. Okay? From the word primer, tagasimula. So, yung unang mga nucleotides lang yung linalagay niya. Okay po? Ayan. So, next. So, ganito yung function ng helicase. So, ina-unzip niya yung strands. Okay? Then, yung RNA primase naman, maglalagay ng initial nucleotides. Ayan, para masimulan yung replication. Ayan, dito na tayo sa next part ng ating replication, which is elongation and polymerization. Okay, once the primer has been added by the primase or the DNA polymerase alpha. Okay, DNA polymerase alpha is just another name for the primase. Okay? So, ayan po. After that, after magdagdag ng primase ng nucleotides, meron naman tayong tinatawag na DNA polymerase 3 na mag-a-act after that. So, ang gagawin ni DNA polymerase 3, mag-i-insert siya ng mga nucleotides dun sa 3' end nung primer. Okay? So, yun ang function niya. Now, we have the leading strand and the lagging strand. Okay po ba? So, yung leading strand, nandito siya. So, leading strand. Lagyan natin ng letter L. Okay, yung leading strand po, ito yan. Itong 5' to 3' direction. Okay? Yan po. The 5' to 3' direction. Yung counterpart naman niya, or yung template niya, is itong 3' to 5'. So, this is the template, the 3' to 5' direction. Okay na po. Ayan. So, ito yung template, ito yung leading strand. Okay, itong leading strand, continuous yung paglalagay ng mga nucleotides ni polymerase 3. Okay? Basta ang keyword natin, pag leading strand, continuously inserting nucleotides, yung polymerase 3. Sa lagging strand naman, discontinuous or fragmented yung paglalagay ng mga nucleotides. Okay, so later i-explain natin siya. Okay, yung pagkakaiba nung leading strand and lagging strand. Pero later pa, okay? Para smooth yung transition natin. Okay? So dito tayo sa lagging strand muna. Describe muna natin yung lagging strand bago tayo mag-move sa susunod na slide. Yan. Nakalimutan ko. Okay? So, itong lagging strand, meron tayong template din. Okay po. Itong kulay orange, that is the template. Okay? Now, yung kanyang lagging strand ay itong kulay pula. Okay po. Now, yung lagging strand naman, yung direction niya ay papunta sa 3' to 5' direction. Okay. 3' to 5' direction. So, papunta dito yung direction. Okay? So, at least, yun yung pagkakaiba. So, lagging strand, 5' to 3'. Ay, leading strand, rather. Leading strand, 5' to 3'. Lagging strand, 3' to 5'. Yung direction. Okay ba? So, yun yung direction nung paglalagay ng nucleotides. Okay. So, here we have the DNA polymerase inserting nucleotides in the leading strand. As you can see here, it is continuous. Now, before we discuss what happens in the lagging strand, ito muna pala. So, we have different types of polymerases. We have the alpha, delta, epsilon, gamma, beta, and so on and so forth, as you can see here. Okay. For the alpha, ang ginagawa ng mga alpha polymerases is they provide the start. Okay? They are the primers. So, naglalagay sila nung initial na nucleotides na kailangan. Okay? Yung delta naman, itong delta, ang function niya ay para sa lagging strand. Okay? Supplying or inserting the nucleotides in the lagging strand, repairing and proofreading them. Okay? Same with epsilon. But for epsilon, yung area naman niya ay yung leading strand. So same din, synthesis, repair, and proofreading. Now, delta and epsilon are very important because nagpo-proofread and nagre-repair. Kapag nagka-problem sa repair and proofreading, there is a high chance na magkaroon ng harmful mutation. Okay? Now, we have gamma. So, gamma naman, hindi siya nasa nucleus. Instead, mitochondrial DNA yung ginagawa niyang synthesis. Okay? For the beta, we have the base excision. And for the eta, zeta, kappa, iota, theta, lambda, mu, and nu, they are responsible for the DNA repair. So, additional DNA repair mechanisms. Okay? Ayan. Pag-usapan naman natin yung lagging strand. So, sa lagging strand, madaming RNA primase ang gumagalaw. Okay, pero dito sa GIF na makikita nyo, isa pa lang. So, sa next slide na tayo para makita nyo. As you can see here, dun sa previous slide natin, merong in-insert si primase na initial nucleotides. Okay? Now, after that, meron tayong DNA polymerase. Okay? Yung DNA polymerase na yun, mag-i-insert siya ng necessary na nucleotides para masupplyan yung strand na to. Okay? And as I said a while ago, fragmented po yung paglalagay ng nucleotides dito. Okay? As you can see here, itong fragment na to, ang tawag sa kanya ay Okazaki fragment. So it is named after the Japanese scientist who discovered it or who named it. Okay? So ayan, fragmented ang lagging strand. Yun yung tatandaan natin. So para ayos, ito po. So we have the leading strand and the lagging strand here. So makikita nyo dito, ayan, itong leading strand tuloy-tuloy, continuous. Pero ang nasa lagging strand, discontinuous or fragmented. So mga Okazaki fragments yan na tinatawag natin. Okay? Now para maging stable or ma-connect yung mga Okazaki fragments, kailangan natin ng another enzyme, which is called, now, DNA ligase makes the makes the Okazaki fragments and the strands stable again. Okay, by providing chemical bonds that strengthens these sets of nucleotides or fragments. So from fragmented, ginagawang isa ni DNA ligase yung structure. Okay? So yun yung function ng DNA ligase. Okay. Now, let me give you a summary of the elongation and polymerization process. So first, we have helicase. So yung ginagawa ng helicase is unwound and unzip. Okay po. Now we have the SSBS or the single strand binding proteins that stabilize the unwound parental DNA. Okay? And stabilize para hindi ulit magsarado. Okay? Kasi kapag nag-meet ulit or hindi stabilize yung mga nucleotides na yan, ayan, magsasara yung template natin. So, we don't want that to happen. Okay? After that, the primase will act. In this case, we have the RNA primase. So, the RNA primase provides the initial nucleotides, but it was a DNA polymerase who finished the task, at least for the leading strand. Okay? So sa leading strand, ayan, tuloy-tuloy yung paglalagay ng DNA polymerase ng nucleotides. But, sa laging strand naman, madami tayong primase. Okay? So, yung mga primase na yun, syempre, initial or the start. Maglalagay sila ng nucleotides na necessary para sa start. Then, syempre, yung DNA polymerase naman. However, discontinuous ito. So, after a certain numbers of nucleotides, aalis na naman sa DNA polymerase. Okay, to form the Okazaki fragment. Ayan. Now, this fragmented parts will be stabilized by the DNA ligase. As you can see here, the DNA ligase is already acting here. So, in-stabilize na niya yung mga Okazaki fragments dito sa region na to. Okay? So, the DNA ligase stabilize. Okay? So, that's it. That's the gist of the elongation and polymerization. Now, let's talk about the last part, which is termination. This is the last part, which is termination. So, remember the replication forks? So, we have two replication forks, right? So, we have one replication fork here, and the other replication fork here. Now, there will come a time that these replication forks will meet. Okay po. By the time that they meet, ayan, successful na yung elongation and polymerization process. So, by the time that these replication forks meet, these spaces meet, by that time we have already two replicons. Each replicon is made up of one old template and a new complementary strand. Okay? So, yun na. Ganun lang kasimple ang termination. Okay? So, that's it. So, if you have any questions, just email me or just message me via our GC. Okay? So, that's it. Thank you for listening. Okay, pasensya na di ko na-edit ito. I don't have any time to reshoot this again. So, sige. That's it. Thank you for listening. Bye-bye.